quad view so that you can actually see what's going on right now. We're being drugged through the water uh, by expedition vessel Nautilus at the fastest safe speed to move us through the water with both ROV attached and in the water. On satellite feed 2, you're actually looking at the tether that connects Argus to Hercules. So the two lights in the center of the screen are the actual Hercules rover being towed by the tether. On satellite feed 3, that's the opposite view. You're looking up at Argus. Argus is the sole bright light in the center of the screen from the tether as it connects to Hercules. So that's pretty much the state of things as we transit to our next area of interest, which, while only 4 nautical miles away at the speed we're traveling, should take about 3.5 more hours to get there. So unfortunately, this is what we'll be able to show you for the next 3.5 hours, but hopefully the conversation will keep you entertained with the next group. So stick around and we'll see you on the next shift.
Of it.
Now
Hello, is there anybody in our front row? Aside from our pilots and navigator?
I'm here.
We're here.
Okie dokie. So we're doing about a 4-hour midwater trawl, or tow, trawl, midwater tow.
Yeah, we're gonna avoid making it a trawl. Yeah.
Probably a good idea.
Please do. Okay, I'm going to go over some notes from the last shift. Just let me know if you need to hear from Jan.
Is now a good time to address ourselves to the audience so they know who we are?
Sure seems like a great time to me.
Excellent time.
And
Well,
everyone can make really long introductions.
I was gonna say, we might want to wait until we're a little more bored.
we could do a refresher later for our new audience members 2 hours in or something.
Reprise.
Yeah, we'll do introductions every hour.
Yeah.
all right, sounds good to me. Well, good evening or good morning for wherever you are across the world. My name is Brandy Jones, the Science Communication Fellow from A-Leaf ISD, bringing STEM education and excitement to the students there so that they know what career paths they have available to them in the future. And I am super excited. Hey, Miss Cole, I see you here. Thank you for tuning in.
Oh,
Anyone else?
it's back.
Is it my turn?
Yep.
My name is Erin
Wait,
Rainey.
if the intros are that short, we're never gonna make it.
Okay.
I agree.
So Brandy, what school do you
I
what
need
school do you work
details.
for?
Yeah, let's do more
Yeah.
detailed.
Okay, so a little more detail.
And where are you from? And what's the name of the town you live in?
I am from Houston, Texas, and I work for a school called A-Leaf Independent School District. I am the STEM coordinator and the science and technology specialist there. What else is there?
How about some kind of fun fact or question you come up with that all of us have to answer?
A fun fact about me. I'm a twin. I have a twin brother.
Oh, that is a good fact.
that's
Okay.
That's a really good fact.
I don't know what else to share.
That's good for now.
Okay.
We'll let you off the hook.
All right, sounds good. You ready to roll, Miss Erin?
Now it's my turn. My name's Erin Rainey. I am the video engineer for this watch. And if I'm not here, I'm out filming wildlife. Your turn.
Oh, that was really short too. What about a fun fact?
What type of wildlife?
Whatever they send me out to film.
Give us some example of things that you filmed.
I filmed bears and alligators and albatross and penguins.
Lions and tigers?
No, not
Wolves.
lions and tigers.
Not
wolves. I
yet.
have done wolves.
What's the smallest thing that you filmed
The smallest thing I've filmed,
on purpose?
not on accident.
Not on accident.
I was filming these little chicks down in Florida on the springs, and they were walking along. They were kind of behind their moms, and then one stepped on a baby alligator's head,
Oh no!
and then it ate it. And that was the first day on one of my jobs, was filming
my
that.
God.
What kind of chick? Was it a plover?
No, it was oh now I forget what they're called. A moorhen, I think.
Oh,
moorhen.
Did the
moorhen.
alligator eat the chick, or did the chick eat the alligator? It was
No,
unclear.
the chick stepped on the alligator's head, and then it snapped it up and drug
Okay.
it underwater.
Yeah, because the
Yeah.
other way around would be.
That'd be pretty cool.
And
It would be a really big chick.
yeah, it would be.
Your turn.
All right,
Which
I'm
would
up.
go against the initial question.
Hi everyone, my name's Paul. I'm the ROV engineering intern piloting Argus. By day, I'm at UC San Diego studying mechanical engineering and robotics. What else are we going over here?
Fun fact.
Fun fact? Ooh. No, I'm not fun. I don't know.
What was Aaron's fun
Interesting fact.
Wait a minute, wait a minute. What do you
Yeah.
mean you're not fun?
I'm
Yeah, you're a
You were
fun
dancing
guy.
on the tables after dinner. You got all excited about the ice cream that was served, and then you started dancing on the tables.
I was dancing on the tables?
You don't even remember you were in this sugar high, weren't you?
Sugar high blackout.
Yeah. So I've been off sugar for a couple hours now. You know, it's
Withdrawal symptoms.
Withdrawal symptoms.
All right, so Paul's fun fact is that he has sugar blackouts. My name is Greg Diffendale, and I'm the Herc pilot. By day, I'm a Herc pilot.
By night, you're a Herc pilot.
By night,
Apparently
I'm a
by
Herc
night you're a
pilot.
Herc pilot too.
And fun fact is that the favorite animal that I've seen underwater was in the Galapagos. It was the flamboyant squidworm, and beets are my favorite root vegetable.
We're not even asking that question.
But let it be known from now on that is Greg's favorite
not Greg's. He
animal,
doesn't get
and
to
no
take
one
it.
else can ever use
not
that
fair.
for anything ever again. Looks like someone else has to get a new favorite animal.
Is everyone ganging up?
I just figured you'd really appreciate it. Actually, my favorite animal is the, and it was unfortunate that yesterday he was tumbling down a slope, but the bristleworm with the coppery,
that was
Oh
cool.
yeah, yeah.
that's my
That was
favorite.
absolutely phenomenal.
You can
I
have
was
the
thinking,
flamboyant squidworm back.
I was
yeah.
thinking it looked like
Can I
a New Year's Eve dress.
Whoa.
Oh, yeah.
Yeah. Just shimmery and kind of
Wonder why it
flapper
gets like that.
flapper dress looking.
We're moving pretty well.
Is that a float?
Probably.
What are you looking at, Rennie?
Oh, sorry. In the scanning sonar, there's just a single dot, and I thought,
Is it a whale?
no.
No.
I said, is it a float?
Rennie.
One of the floats on the tether.
Oh.
Very small.
Okay.
And just before Rennie goes, my favorite root vegetable is actually celery root, which I've only
Okay.
had once, but was excellent. It was in a hash.
Oh yeah, I've had you like the shaved celery root. I've had that.
It was cut up.
I've had it like shaved, and it
Nicole made it. It was wonderful.
sounds really good. Renato Kane, the navigator on this watch. I sometimes serve as the mapping coordinator, sometimes as the lead navigator. I do a lot of travel when I'm off the ship. Aside from that, I'm on the ship doing this. My day job is this.
Wow, Rennie, that was
Fun fact.
inspiring.
What's your favorite animal?
Oh,
Not the-
George calling.
George Collin?
No, George was calling me.
Oh,
Sorry.
sorry. Okay. George is the mate.
Yeah.
It's kind of important if you talk to him.
Yeah, George. We can only. How's the boat driving go? Sorry, what was the? What are we saying?
Your fun fact or favorite animal or both?
Fun fact. Not very fun in general. So I'll go with animal. Still the deep staria jelly. Yeah.
Jamie, I think it's your turn.
Okay,
yeah, Jamie,
we're
if,
gonna.
if yours isn't exhilarating, then
It's gonna be great, Rennie. You're gonna love it.
Okay.
I'm Jamie Wagner. I am the data logger for this watch, and I'm also the science manager on board, which means I help with making sure all the notes get recorded from different dives, and all these samples get processed when the ROV comes up, and that we type up lots of different information about everything that's happened. And my interesting / fun fact is that I recently got some ducks, and we found out they're both girls, because when you get young ducks, you can't tell if it's a girl or a boy, and they're laying really nice duck eggs. And
Do you eat the duck eggs?
we do eat the duck eggs.
Oh.
You can eat duck eggs just like you would eat chicken eggs. They're good for just about anything except things that you have to whip up to a point. So like not good necessarily for a souffle, though we did make a duck souffle. It just wasn't quite as fluffy. But yeah, you can put them in your baking. You can make an omelet with them. You can make a quiche with them. You can do anything that you can do a chicken egg with, basically. And they're more flavorful and more full of protein than chicken eggs, so they're really good for you.
What kind of duck? Is it a mallard or just a regular domesticated duck?
We have Rouen ducks, which look just like mallard ducks, except they're bigger.
Oh. Okay.
I'm Gary Williams. I'm a curator and coral biologist at the California Academy of Sciences in San Francisco. And let's see, fun facts. Somebody mentioned Galapagos. One time diving in the Galapagos, two things came together that were quite unusual. One was an iguana, a big lizard at about 30 feet underwater, eating algae, and a torpedo-like object going by turns out to be a penguin, a Galapagos penguin.
Oh
So
yeah, I swam with those penguins
Isn't that
too when I was
amazing?
down
Yeah.
there. They're
Yeah.
incredible.
Oh, that's
Yeah,
really
they are
cool.
fun.
Did
The
you
little
say
tiny things.
the iguana was 30 feet long?
30 feet underwater.
Oh, 30 feet underwater. Got my facts mixed up. I was like, that is
If you want to believe it,
enormous.
if
Really
you want to believe it was
both are
30 feet,
impressive,
that's
really.
fine.
Well, if it's 30 feet underwater, wouldn't it still be 30 feet on land?
No.
It was
It's one of those, you know, grow to size
It was
things.
subtitle, let's put it that way. But it was eating algae off of rocks, and you don't usually see lizards eating algae off of rocks in the ocean. So.
That's amazing.
Very cool.
That's very cool. So
Yeah.
I'm Jan Roletto, and I am the dive lead and one of the co-PIs for this project. My day job is I work for the Greater Farallones National Marine Sanctuary, doing things like this. And I tell people all the time, I'm a research coordinator, not a researcher. And yet I get thrown into doing research, which is why I stay in this job. I think I've been in this job for 28 years now. 27 years? Okay, whatever. Fun fact. I have sat on top of a live humpback whale
What?
Wow.
and covered it with towels because it was stranded in the mud in San Francisco Bay. And we refloated it, and it went out to sea after that.
That's a pretty good fun fact.
Is that a pretty good fun fact?
That's
I
such
was
a
trying to-
happy story.
You out fun
okay.
fact us.
And Jan, you have a guest on the live feed.
Oh.
And guess what he wants me to tell you?
Uh,
He says that Alex is not his favorite animal today.
I know why, too.
And Gromit would still like to know if there really is chicken of the sea, and if so, can she get him some?
So, well, I think I'd return that question and answer over to Jamie because she knows the critters of the sea better than I do.
I think chicken of the sea is what tuna is sometimes called.
Yes,
Yeah.
I believe so. But is it really chicken?
No,
or is it
it's
really tuna?
it's really tuna. It's a fish,
Hi,
not a bird.
John. Hi, Gromit.
I love how matter-of-fact Jamie is. It is a fish.
I have to take my science questions seriously.
Yes. And John does not. And thank you, Brandy, for sending that along.
Yes, happy anniversary.
Happy anniversary, John.
You're so sweet.
We'll be celebrating it next weekend. Okay, so
Uh-oh.
what are we looking at in our water column?
A lot of water.
Well, I'm glad you asked, Jan. We've seen some interesting things float by. There's marine snow, which we could have a whole conversation about, but we'll save that for a little while. A big siphonophore floated by recently. It looked like a long chain. It had a reddish section, and occasionally there's a fish. There's one in Argus Cam right now. There's a little red shrimp swimming by the Herc view.
Oh, that's nice, yeah.
Oh, we've also been seeing some ctenophores occasionally. I know there were a lot when we were down on the seafloor, and if you're lucky, you'll catch one floating by up in the water column as well.
Yeah, those ctenophores that we were seeing near the bottom, they were purple, and I've never... Were those ctenophores, or were those, what are the salps called? I'm looking at Gary.
Oh, you mean those chains of salves?
No, the purple. I want to call it a
Purple
salp.
transparent.
Purple transparent salp with the shell in it with the wings.
Oh, pteropods.
Pteropods.
Those are
thank
swimming
you.
mollusks.
Yeah.
They're mollusks. They're not
But I've
salps.
never seen them. I've never seen them be purple in color.
Yeah, I don't know. The way, I guess the lighting was different on that. It wasn't direct on it. It was more of a shadow or a silhouette.
There were
wasn't
several
it?
that were purple. Jamie, do you recall those?
Deep water pteropods, I don't know.
This dive or the previous dive?
Previous dive.
I think it was the previous dive.
Yeah. And they're usually in the shallow water, and I haven't seen any as deep as we saw them. That was,
Well,
what, 2,000 meters?
We
or?
saw one pteropod swim by the camera. I only recall one doing that, though.
I know I saw one yesterday.
We also did see a really dark purple ctenophore go by.
Oh, maybe that was a ctenophore. So maybe it was the lighting, because they were both the same kind of color.
I don't recall the color. I don't recall the color of the pteropod.
Uh-huh.
I remember it was dark, and that's all. I saw it swim by, but it just looked like a dark object with wings, a cone-shaped shell, and wings flapping around in the water.
Okay.
Yeah. Which is unusual. You usually don't see pteropods.
But I got to say that bristleworm.
I loved the bristleworm
That
with
was
the goldish copper bristles that were very shiny.
I've never seen any animal basically reflect that color.
Does it swam by the camera?
It
Or
sort
were you
of
following it?
tumbled.
It's
It tumbled by.
down,
It looked like there
yeah.
was a strong current sort of blowing it.
Yeah.
Mm.
It started crawling and then it fell off the face and then it started tumbling down. And then it kind of stuck. And once it stopped tumbling, it wasn't nearly as reflective. The bristles weren't nearly as reflective.
So the iridescence was from the reflected light from the ROV then?
I think so.
Yeah.
Yeah, it wasn't generating
wasn't
its own light.
bioluminescence,
No,
it
it
was
wasn't
No.
bioluminescence.
It was iridescent.
I think it was less shiny when it stopped tumbling because a little bit of sediment had been kicked up.
Mm-hmm. Yeah, that's a little jellyfish. How many samples are in the bio boxes?
Oh, that is a great question that I am well prepared to answer. We have 5 samples so far this watch. Our first sample that we took was a push core.
Yes.
And
yeah,
then
we're using those to look at not really climate change, but just some climatology and paleontology, paleoclimate. Did I pronounce that right? Paleoclimate?
yeah.
Indicators in the sediment. So we're taking a series of push cores throughout this cruise from deep to shallow. And Dr. Tessa Hill at Bodega Marine Lab, which is part of the UC California Davis system. And her students are always looking at climate change impacts, especially ocean acidification. So we have that one core from this dive. We have 5 cores from yesterday's dive, is that correct? Or do we only have 4 cores from yesterday's dive? 4.
Actually, we have 3 because one of them didn't really retain any sediment.
Oh, okay. So we have 3 from Cordell Bank area, and we have 1 so far from northern Greater Farallones area. Oh, okay. What else do we have?
Our next sample was a black coral.
Oh, that one was beautiful. I was
yeah.
downstairs looking at that being collected.
And it says it had a brownish, greenish organism growing on it.
Uh-huh.
I didn't see that, so I don't know what it was.
I was kind of moving in and out of the lounge, the room where the monitors are, trying to get these new start and end points kind of situated. So I
guess
didn't
we'll
get to look at it
we'll
a lot.
find out when it arrives on deck later.
Yeah, but some of these species are species that we don't know of from the area that have been requested by other scientists, including Gary and his team at California Academy of Sciences. But other ones are for species inventory for either Cordell Banks Sanctuary or Greater Farallones Sanctuary. So we have 1 black coral with an unusual. Do we know what kind of like the genera of that? Parantopathes for now.
Parantopathes?
Parantopathes is the black coral.
Oh, okay. I thought you were asking about the other organism.
Yeah.
That was on
No, I don't. I didn't get to see closely that other organism. I knew it was there, but
Is it on? Oh, the front page is missing.
no. This is the one that has the front page. And so what else do we have?
Our next sample was a Niskin water sample that we took right next to that black coral sample.
Who's talking?
Oh. Brandy, are you saying something? Oh, yeah.
I think that was Aaron.
Here's the parentopathy.
How's that, Aaron? And plus, I'll try and remember to speak louder. Okay. So we have a paranthropathes, and
then
we have a Niskin bottle, and the Niskin bottle is for a couple of different projects. The water samples that we get in the Niskin bottles are for specifically 2 different projects, but so we only need 1 sample for these 2 projects at different depths.
Jen,
And
someone
yes.
on our live feed would like to know, what does the suffix pathes mean?
I can't hear you.
Someone from the live feed would like to know, what does the suffix pathes mean?
Packy, that's a good question for Gary.
Yeah, antipathes, that's the word they use for the black corals, because in the old days they thought that if you took the medication made from the ground up to axis, it would reduce disease. So pathes means disease. It's kind of like pathology, the study of disease. That's where the word comes from, meaning disease.
Nice.
I didn't know that before. That's a great little fact.
Yeah. So a
Antipathes,
black coral that
antipathes.
has the name antipathes is anti-disease. Okay.
It's a remedy for disease, back in the medieval days anyway.
And black corals are considered black corals because the skeletal structure is black, correct?
Yes, it's a protein with a lot of sulfur in it. So like if you had your fingernail with a lot of sulfur in it, it would be black. So it's basically the same thing as your fingernail, but it's black. So that's where the name comes from. But the living tissue doesn't look black at all. It's usually green or brown or golden color.
Yeah. And it's really hard to see on camera unless you get the right lighting and you get in really close, because it's covered by living tissue.
That's right.
Yeah. All right. So we have another coral sample, right?
Yeah, so our last sample, so we only have 4 samples. Our last one is a light pink Peristinella coral.
Oh, yeah. That was pretty common, but that was a new inventory species for Greater Fairlawn Sanctuary. We did see it in Cordell Bank, pretty far north. Gary, do you know what the range of that would be? Is it still within its range? Yeah.
Ramosa, if it's Peristinella ramosa, then I think the range is basically California and maybe up into Alaska, but that's about it.
Oh, okay.
But the genus is, I think, almost worldwide. It's common. The genus Peristinella.
All right. So we have plenty of space in our bio boxes and our Niskin bottles and our push cores for the second half of this dive.
Yes, we do.
And yeah, so we keep a lot of students busy. Ooh, what we got there? Looks like we got a little bit of a jelly piece that got caught in the motor.
Jelly pieces.
This might be a good question for the ROV team. Do you guys ever play video games? They'd like to know, do you play video games?
Greg?
Not regularly.
No.
Let's see, I have whatever the second generation of Xbox was. But I think I only have like 3 games for it.
Go ahead, Bridge.
Cool.
It's
Yes, Jamie. Hey, Greg, I'm noticing that the porch is a bit brighter than it has been. Did something change with the lights?
no.
Oh, well, I guess it's just me then.
Yeah, we angled the down LEDs a little bit more, just to because we kept pushing past things and getting them into the shadow. So we're giving this a shot, and we probably will move them back a little bit. If we could get the light pool to kind of hit not
The nozzle?
that bar.
Yeah.
Well, the nozzle's okay, but like the first few inches of the porch would be fine.
Yeah.
But it kind of is like the porch LED is on, right?
But that's not...
Kind of washes out a little bit.
So down is... Does...
Those guys.
Right, but forward is for farther afield, right?
Yeah.
So I guess we'll have to see effectively... you didn't change those, though, did you?
No. But that one.
It did...
As you can see the little circle.
Yeah.
Ready, and boom.
Oh, okay. Now, was down and forward, did they meet, and are we sacrificing any light out?
Like in between? I
Yeah.
don't think we're sacrificing any light in between.
Yeah.
Where you going?
Are you following in, Bubble? It's looking right at us.
Oh,
Go ahead. 1.5.
I can't be good for it.
Yeah, I think 1.5. I'll talk to Greg about that.
Greg, you should have let
Want to
me
go any
go for
faster than
that.
1.5?
How are they doing? I mean, it looks like they're redlining, but
Yeah. You want to try for 2? I mean, there's not tension on the winch. It seems to be okay.
yeah, we can go for 2. We used to do 2 before, so.
So,
Bridge nav. Can you make 2, do you think? 2 knots?
ooh.
Okay. All right, I'll come up and help you.
The back row will get out and we'll use
Rennie's
oars.
getting the oars out.
Yeah. So who
I didn't
controls who was controlling the camera? Was that Aaron or was that Greg?
I control the
Uh-huh.
pan. Well, that was the.
Okay.
That's
I control the
great.
pan
And then I
and the
control
tilt,
the zoom.
and
So
Aaron
that
controls the zoom.
that one image where you could see the lasers coming out from on top of the camera, could you do that again? I'd like
Oh,
So yeah, so that's Paul. Paul can controls the bubble camera.
Okay.
is the bubble camera
That
up
is
on?
really cool. Because now I think people will have a good understanding of the lens and where those lasers that we always see from Herc.
Is that up on the quad?
Wait, hold on. I'll put it up on the quad. Yep, there it is.
It's on channel 2.
Oh, so it's on channel 2. That is, I think that's very
Just
interesting.
for a second.
Just from a,
So for everyone
how does the
watching,
ROV work?
Yeah.
over on channel 2, we're showing the lasers that are on top of the main HD camera.
And they're actually parallel.
Yep.
And so they're actually set at 10 centimeters apart.
Yep.
And we use them to measure things. So before we collect, specimens, we'll shine the lasers on the specimen if it's large enough, and if it's too small, like a C-pen, we'll shine the lasers on the seafloor, but right next to the base. Because if you shine it further away, you're not going to get an accurate measurement.
Correct. Cool. Yes. Played this game before. Same camera, 10 years.
We have another question in the last
So I
feed.
think Dan said on his way out we're shooting for a depth of 500 meters.
Yep.
Okay.
Yeah.
So we're getting there.
Has the ROV team ever experienced an unexplainable weird event or even seen like a weird object?
Unexplainable weird event. Yeah. Let's see. Well, that one was explainable, but last time Aaron and I were doing a tow, the screens all went dark. One of the high voltage copper lines on the legs on our 3-phase conductor in the cable running down to Argus shorted to a stainless steel tube, grounded tube in the cable. And so our ground fault sensor tripped and shut off the vehicles. And luckily that was the end of the last dive at the end of the season.
Wow.
So that was pretty good. But that one's explainable. Let's see. I've seen a soccer ball about 70 kilometers away from any bit of land, just on the seafloor.
Wow.
Saw
Soccer ball.
a Coke can from like every decade for the past there are like 6 different types of Coke cans all in the same depression.
Oh, wow. That is odd.
Yeah, it was just really kind of like strange.
Where was that?
I was somewhere in the Mediterranean.
Oh.
I've seen all kinds of weird trash, toilet, refrigerator.
Toilet.
I'm surprised how much, debris, man-made objects, human-made objects were on the seafloor this morning.
Out of sight, out of mind.
Yeah.
I saw the Ziploc bag.
Yeah.
underneath one of
We're
the
typically not
sponges.
that trashy around here. It's like, wow, there's that. Oh, well, look at that. Oh, there's like a square door or something, some kind of panel.
Yep.
A tire.
So are those coming from shore or getting washed in from further away?
Just because there was so much in a small area, and pretty much our seafloor in this area, and I only really know the seafloor in this area, really is not that trashy. So I was thinking that maybe somehow something got dumped overboard, either intentionally or unintentionally, kind of scattered it along in that one area. But it's pretty deep.
It's hard because it's hard to enforce, right? The dumping of waste, especially on a global scale.
Yeah, but the looks of it it didn't look like it was
Yeah,
like
that
a
seemed pretty
dump.
local.
Yeah, it looked pretty local, and so most likely I would think that would be accidental. But still, it was little jelly. So I was surprised to see that much kind of trash or debris that far offshore and that deep off of California. Jamie, are you on the next cruise?
I am.
You are. And then that's the last cruise of the season, right?
Correct.
Let's see, Jamie, Aaron, Paul, myself, Rennie, we're all on the next cruise.
Is Nicole going out on the next cruise too?
Yep.
Oh, okay. Good team.
Mm-hmm.
Yep.
Should be a good effort. Chad and his sanctuary team down in Monterey Bay National Marine Sanctuary. Go back to the octopus's garden.
Yeah, that should be neat.
So that wasn't on Davidson Seamount. That was kind of not on the Seamount itself. Is that correct?
I was not on that cruise. So
I wasn't, yeah.
I was on that cruise, but I don't remember exactly where it was.
I think it was just maybe, I don't know. I think it was near Davidson Seamount, but not on it.
Yeah, it actually, to follow up what Jamie said, it becomes kind of hard to separate different things. Years, oceans. It's like, oh yeah, you were there for that, and they're like, no, I was not. And they're like, oh, that was only a couple years ago, but it was in the Med. And like, well, we haven't been in the Med since 2012, so.
We're at a depth of 500 now.
Yeah, let's go ahead and stop.
We have Miss Cole from Houston, Texas, and Ailey FISD wanting to know what areas have you noticed has the most debris underwater?
Ooh.
Well, I'm only a local gal, so I'll turn that over to you ocean explorers throughout the world.
Let's see, I would have to probably say oftentimes it's like a closed body of water. The Gulf of California and had some surprising bits of trash. The Mediterranean also. But again, it wasn't strewn everywhere. Usually it's like just lots of little bits.
Yeah, I think I recall seeing a fair amount, again, not strewn everywhere, but little bits here and there. On off of Los Angeles, we were diving really close to a shipping lane.
Oh,
So
yeah.
that's a really high traffic area, and we were seeing a fair amount of man-made garbage down there and some old fishing lines and nets and that type of stuff as well.
Yeah.
And in truth, probably some of the, and it wasn't on the seafloor, but all of the Pacific Islands that I have been to, the uninhabited Pacific Islands, have just trash all over the beaches. Anything that floats ends up, you know, there.
Yeah.
One of them. And it was just amazing the amount of trash that was on an uninhabited island.
Wow.
We had that in Alaska too. We were on an uninhabited island, and it was in an area where the currents come. And within just arm's reach, you could pick up 50 water bottles
Yeah.
along the beach,
it's
and it
everywhere.
was pretty eye-opening. I mean, you see it in other places where there are tons of people, but to see it somewhere where like the animals on the island have never seen people, but they see us and, you know, garbage and all of that. Yeah, it makes you never want to use a water bottle again.
Yeah. Is it hard to determine or differentiate between debris and marine snow? Like right now, we're looking at, is this marine snow or
This
is
is
it
all,
debris?
this is all
This is
organic.
all dirt.
Okay.
Organic. It's all natural. Yeah.
All dirt.
Gary, what's your next project? I know you sometimes go to the Coral Triangle in the South Pacific.
Yes, that's exactly what we do. Indian Ocean is where we want to explore next for coral reef surveys, shallow water coral reef biodiversity surveys in the Indian Ocean.
Wow. Do you dive or do you snorkel when you do that?
Scuba diving.
Scuba diving?
Mm-hmm.
Wow. And has Cal Academy done expeditions in the Indian Ocean before, or is this something new?
Yes, we've in Zanzibar, which is in Tanzania and East Africa, and the Maldives and Reunion Island, Madagascar.
Madagascar.
And Mauritius. There's an island called Mauritius, which is near Reunion Island. All that area in the western Indian Ocean is relatively unexplored for that kind of research.
That is cool. So California Academy of Sciences, do they sponsor these expeditions, or are they co-sponsored with other agencies, partnerships?
Yeah, sometimes it's National
institutions?
Science Foundation, and we have a coral reef initiative that is sponsors a lot of expeditions, and so it's a combination of different funding organizations.
Good.
And if we're lucky, we get the funding.
Yeah.
And if the permits are coming through from the foreign countries, sometimes you don't get the permits you want, so you can't go. The collecting permits don't come in time, and you have to be there at a certain time of year for the weather and all that, so it just depends. Sometimes you have to cancel an expedition just because of the lack of permits that you're getting from the country, the host country.
Yeah.
So since we have the time, if this light's on, then means basically the controller is on, the arm is on. It's holding position now. If that light's on, means you can move this around however you want. Arm's not going to do anything. As soon as I hit this button, if I move this around, arm's going to move.
For those of you who are tuning in, this is Hercules, one
So
of our ROVs.
most people pilot with this straight. Originally I learned with it bent up at a 90, but there are very tiny wires in here, and so you don't want to go back and forth a bunch. So I adapted because most of the time it was me and two people who like to pilot it with it like this. Some people even pilot like that, which is a full-on flute player. But the real thing is that, you know, if you think you've hit the button but you can't see the light, you just do a little bit of movement, right? You know, the last thing you want is to do something like that and just have the arm go flying. But yeah, so each joint does something. So, right, wrist yaw, wrist pitch, and let's go ahead and move out. Elbow, shoulder, and then shoulder rotate, your shoulder azimuth. And then you just have a trigger here for closing. Now,
And wrist spin.
and you have your wrist spin. There's a couple of different types of wrist spin, but before we get into that, so there's 3 buttons up here. This one's your index button. You can also watch down here, right? It says index, so I can move this. If I tap it, it says engaged. Now if I move this, the arm's going to move. So if you can't see the button, that's another place you can look for confirmation that you're safe to move. The other 2 buttons up here is wrist control. Now right now, it's set, so if I go like this, I'm going to rotate my wrist. Now you don't have complete motion with the wrist, but you can get a lot of stuff, and I find it's more natural. Not everybody follows it. A lot of people like to have it, so this does nothing, but you use the rocker